Okay, guys, so for a lab today, we're going to finish up the last, all but the last slide for the study guide. So I thought I'd share that. And as we did last time, I pointed some of these models out, but I didn't really talk about the what's going on with the liver and the pancreas. So this is going a bit ahead of our lecture. And in fact, this is the lecture that I didn't have the video working very well, so I'm going to be straightening out. So it's probably worth going through these figures in the lab just to make sure you know what you need to study for the lab test. But it will help you with the last few essay questions too. So my dog is talking to me, so you might hear him in the background. So I believe I went through, and we're going to be talking about most of these structures. I went through last time and I X'd out a few things that you don't need to know. But the main points that I want you to focus in on are not just what are these things, but what regulates them. So in lecture today, we learned a little bit about CCK. There's another hormone called secretin. And secretin is released, again, these are both released by this duodenum when food arrives. Now, the neat thing about secretin is it gets stimulated by low pH. So cells that align this in beginning part of the duodenum are going to, of course, see low pH regardless of what you are eating. So some secretin will be released regardless. Now, the neat thing about secretin is it is going to solve its own problem, as it were, because what it tends to stimulate is the release of bicarbonate-rich solutions, including bile. So I mentioned with the alkaline tide in lecture that some of that blood that is coming in the hepatic portal to the liver will be very alkaline rich with lots of bicarbonate in it. That bicarbonate's picked up, made into bile, and that's stored in the gallbladder. There are other cells that you're going to be drawing today that are in the pancreas itself, and they form the lining of the duct, at least near the site where the enzymes are made. They are these ductal cells that make a bicarbonate-rich watery solution as well. So the ductal cells are going to secrete their bicarbonate-rich secretion into this duct. Bile with bicarbonate-rich solution is going to be stimulated to be released into this duct. They join together and then simultaneously are released into the duodenum. So secretin kind of solves its own problem because it causes these cells to release the ductal cells to release bicarbonate, and the bile arriving will be rich in bicarbonate and therefore help neutralize that pH. The other thing you drew already is the Brunner's glands that are also stimulated to release their bicarbonate rich solution. The other thing that secretin does is it stimulates the release of CCK, and CCK stimulates the release of secretin. So these two guys talk to each other and amplify each other. But the reason CCK is primarily stimulated is because of the arrival of fat and protein in the duodenum from the stomach. So that CCK is going to go and cause not only cause contraction of the gallbladder and therefore cause bile to come down so that you're able to digest your fat, but it's going to talk to the other cells you're going to draw today called the Cynar cells. These cells release the enzymes, including protease precursors and inactive proteases and lipases. And those will come down these ducts, again, the bile, and the enzymes arriving at the same time, but they can't leave until this sphincter opens. And the sphincter I mentioned last time is sometimes called the sphincter of Oddi, which is fine by me. It's a lot shorter to write than a hepatopancreatic sphincter. Don't worry about the ampulla. So that way what you get is CCK and secretin working together to cause both bile, which is bicarbonate rich, and the pancreatic solution secretions, which are enzyme rich and bicarbonate rich, to all arrive in the duodenum when the food arrives. So if you have protein and fat in your diet, you're going to get maximal amounts of these guys arriving. Now, there are some other effects of these hormones that we'll talk about in lecture, but sometimes I will point at, the sphincter and say, give me a signal that would cause that sphincter to relax and open, and then you're looking for CCK in that case. Or I point at the acinar cells and say, what hormone will cause greater release of enzymes by this cell, and then you would tell me it's CCK again. Okay, so let's talk a bit more about these various ducts that carry bile. So when the liver makes the bile, it makes it already mixed with a bicarbonate-rich solution, but it's in a more dilute form. The gallbladder itself is going to have two functions. One function is going to be to store and then cause a contraction of the gallbladder and therefore empty the gallbladder, and a surge of bile come down and go into your duodenum, assuming CCK opens a sphincter. But the other is that as bile sits in the gallbladder, water is removed and it becomes a more concentrated form. So if someone loses their gallbladder, has to have it removed, they still can get bile, but the bile that arrives will be not as concentrated, and it won't be reflexively a big pulse of bile that's squirted down when CCK levels rise. So they're not going to be able to digest their fats quite as efficiently as somebody with a gallbladder, but they can digest fat. They have to be careful on what fat is in their diet. The other thing interesting about it, and I know you have most of you have not listened to the lecture, is that this sphincter, like all the sphincters we've talked about so far, is in its default mode, it's closed. So when you aren't causing CCK to be released, or even secretin to be released, the liver is making bile in kind of a constant way. It's entering into these right and left hepatic ducts, and from there flowing into the common hepatic duct. So common means it comes from two sources. Hepatic, it's liver, two sources of the liver. It flows down towards the sphincter, but the sphincter is closed. So it's continually making this, but as it's making it, it's filling up this duct. So there's always bile in this common bile duct. That bile backs up, actually, as long as the sphincter is closed, and ends up just by a backward pressure going up the cystic duct here and concentrating or ending up in the gallbladder to be stored and concentrated. When CCK arrives, therefore, the bile goes out the cystic duct and then down the common bile duct to be ejected into the duodenum. So this is kind of a two-way road in here, where bile both fills the gallbladder by backing up, and it all depends on the closure of that sphincter to do that. And then when you go to release bile will come surging out through that same common bile duct and cystic duct. Okay, so some of that, we're going to take a look at the models again in the cadaver here. So let me see. So here we have the gallbladder and the cystic duct coming down. And I probably in this figure couldn't tell you where the common bile duct began and the hepatic duct. I don't see them here. So probably the only thing I'd be able to ask here clearly would be the cystic duct. I know this is labeled bile duct, but I can't see where the hepatic ducts are coming in, so I don't expect you to see it either. You do see the main pancreatic duct, and then, of course, just recognizing the organs. This is the duodenum that's been dissected open, so you can see the circular folds in here. There's something else I could ask. Otherwise, the round ligament I mentioned, this is the one that goes to your umbilicus, and in a fetus has the umbilical vessels in it, but then later as an adult, sorry, the bird is going nuts, it is just helping suspend the liver, which is a really heavy organ. I know they point at the common hepatic duct. I think it's pretty hard to see in here. So I have so much better models pictures or models to point at those things. I probably wouldn't use this figure for that duct. Okay, so here is the pancreas I just mentioned. I suppose, say, name two cells that form this, and then you'll name the acinar cells that you're going to be labeling that make the enzymes, and the ductal cells. There's also those would be exocrine cells with ducted glands, and then there's actually more than two, but there's two that you're learning, endocrine cells, which are from 103A, the islets of the pancreatic islets or islets of Langerhans, which we're going to take a look at in the slides in a couple minutes. Now, those don't secrete into these ducts. They're endocrine. They secrete into the blood. So I mentioned right here, and I'm not sure why that was never painted white, but that main duct comes in at the same place that the common bile duct comes in. So right there would be the sphincter of Oddi. Behind this is the stomach, and then this is the duodenum. So I did make these as labeled slides so that you could see them. So here's one of the versions of the liver. Once again, the additions that you didn't see before would be the two blood vessels on this, the hepatic artery and the portal vein over here, coming from all the other digestive organs, the stomach, bringing that bicarbonate-rich blood, bringing the nutrient-rich blood from the duodenum and the other parts of the intestine and the large intestine through those mesenterics, you might remember. And then they only show the common hepatic duct, the common bile duct, and the cystic duct here. They don't show the right and left hepatic ducts. But this model does show you the right and left hepatic ducts. And then a really short, let me see if I can turn this thing into something smaller, a really short, like just from here to here is the common hepatic duct, and then cystic duct and cut off very short again is the common bile duct, just the stalk of it at the end there is shown. This also highlights the falciform ligament, which again adheres to the mesentery around this, so it helps hold the liver in place. It does divide the lobes, but I don't typically ask you about the lobes. And this has the portal vein. I don't know if that maybe is red and is supposed to be the portal artery, but I didn't label it because I thought the other picture showed it a lot better. This is the vena cava. So those are the models of these structures. Now let's go into the liver. We're going to cut a cross section through the liver. And you'll see that the liver is made up of repeating groups of cells that are surrounded by connective tissue. Those groups of cells are called lobules. So it's almost like building blocks or bricks that are put together in a very organized way. Now, they make them look hexagonal in the pictures in the book. They don't have to be hexagonal, but they look kind of like they have straight edges. So I have to go to the mouse now. So this is a really low power picture. You'll see in the slides that are loaded, there's a bunch of low power pictures. In this particular one, it's near the edge of the liver. The stain here has stained the connective tissue, the collagen, blue. So that's why you're seeing these lobules so nicely outlined. So this is made up of many, many hepatocytes, plus the blood vessels and the ductal cells. And all the, let me do it again with the drawing here. All the bile that is made is going to be going through ducts and heading towards the edge. So I'll put a B on this. And that's going through a series of ducts. Now, the ductal lining are so thin, there's no way I would be able to point at anything or ask you to label anything that are the ductal cells out here. But what happens instead is they form larger ducts in this connective tissue. So we're going to eventually look for a bile duct out here. Now, being a duct, it's lined with cuboidal cells. If it were a blood vessel, the lining would be simple squamous epithelial cells. But ducts have cuboidal cells lining them. So we'll go out here eventually and look for those ducts. Now, two types of blood flows. This is bile. Two types of blood flows into these lobules. And one type is nutrient-rich, but oxygen poor, and it's coming from the portal system. The other type that's flowing in is from an artery, and it's oxygen rich. I don't know about the nutrients, but we'll just put an A there for artery. So these two types of blood don't go in parallel veins, kind of like I made it. They go into a capillary bed that is a common capillary. So they're going to fuse together into a common capillary bed. And this capillary is sinusoidal because you're not only going to be putting like the proteins that these hepatocytes make, the albumin and the fibrogen and all that in there, but you're also going to be recycling red blood cells. So remember, the liver is part of what takes up the old dead and dying red blood cells and breaks them down and then stores their iron and all those things you learned in blood. So this is a sinusoidal capillary. So probably the cracks you're seeing here are those sinusoidal capillaries, but hard to say because it could be the duct openings too. So that eventually this combined oxygen-rich and nutrient-rich blood flows through these capillaries, and then ultimately it drains into this large vessel in the middle of each lobule. This is called the central vein. That central vein eventually drains out through the hepatic vein to the vena cava and communicates with the rest of the bloodstream that way. So I'm going through this because of a structure that we're going to be looking for out here in the connective tissue called the triad. But also I want to take a little closer look at this central vein. Now there is blood in this, and they did that kind of on purpose. That's blood that's in the middle that you're seeing in the middle there. So maybe let's go to the mouse and go to the next one. So here is that, and I did have to erase everything, I guess. Well, it's actually not a bad drawing for here. Here is that central vein with blood in it. You can see a little bit of the connective tissue around it's blue because it's a larger vessel. And all these cracks are either sinusoidal capillaries or ducts. It'd be kind of hard to tell. So what could I ask on this? Well, these cells that are making it up, the light pink purpley cells, those are called hepatocytes, and they're extremely busy cells. So SA5 is all about all the various things these cells are doing. So very metabolically active, responding to hormones, making blood proteins, making immune components, and they're just busy cells. They're a great review, as I mentioned in lecture. This slide, I don't ask any structures. It's a different version. There's no blood. That's the central vein there. But what happened is they fed this animal some probably India ink, which is just basically carbon particles suspended in water. And it got taken up, the little carbon particles. And then these black things are macrophages that are taking things that that are foreign out of the portal blood that's coming in from the digestive tract, and it's just using the India ink or the carbon particles to highlight how many of these macrophages there are. So, of course, if you absorb a pathogen through your duodenum and it goes into your portal circulation, you don't want it to go to the rest of the body. So the liver also plays a big barrier role by having these fixed macrophages that are going to remove things that are foreign from your what you absorbed into your bloodstream. So those two pictures focus on the central vein, the hepatocytes, and as I mentioned, these fixed macrophages. They actually have a name, Kuffler's macrophages, but I don't typically use it because it's one of those people's names. So over here is a drawing from your lab book and your textbook showing that central vein and the portal and arterial blood mixing here in the sinusoidal capillaries. And then the green are the bile ducts that you're going to be looking for. Now, I wish that there was one of these at each corner and it was always hexagonal like this, but of course, that's not what happens. If we were in person, you'd be searching and searching to find one of these structures. There's many of them. They're just not located nice and systematically at the corners like this. Again, the blue is staining the collagen, so it's the connective tissue, but it also gets picked up. I'm going to make it a little bigger here if I can. I guess the only way to do that is to do slideshow, if I can get this over here, and then play from the current slide. And we can go to the actual pictures, but right here, you see this kind of purple circular structure, and it's got nuclei that are lining the edges of it. That is an arterial. That's one of the oxygen-rich blood sources that's coming in. Where you're seeing cuboidal cells like here or here, those are bile ducts. So if it's round, which arterioles are here, and it's not lined by cuboidal cells, then it's an arterial. If it is lined by cuboidal cells and it's round, like these two things, or more or less round, then it's a bile duct. So this thing with the pointer, which is typically is quite a bit bigger, is a vein. It's full of blood, so it's a little hard to see what's along the edge. But you can see along here, these are definitely not cuboidal. I don't see anything cuboidal looking along the edge. So this is the portal vein. Now, for reasons that I don't understand, they call this a portal arterial, but Maybe they call it that because the blood gets mixed together in the capillary beds that come from the portal vein and the arterial blood that is not a portal circulation, but you can either call this hepatic arterial, or you could call it hepatic portal arterial. I don't care, but it is labeled here as a portal arterial. These are, again, are hepatocytes, and these are probably the sinusoidal capillaries because I don't see cuboidal cells, but the cuboidal cells in the ducts are pretty flat, so I wouldn't want to guarantee anything on that. So that's what you are labeling on this. This is called a triad, and you'll say, why triad? There's 4 things here. Well, there's 3 different types. There's the portal vein, the arterial, and the bile ducts. So ultimately, the bile in these bile ducts are going to travel to that right or left hepatic duct, and then down to the sphincter, back up into the cystic duct, and fill the gallbladder. All right, let me escape this. Well, I guess we don't have to escape this. We're going to move over to the pancreas. And this is actually a photograph from your lab book, because I don't, in our slides, easily see any of these pancreatic ducts. I don't have you label them. So if I were to ask this, it'd probably be with this kind of with this picture, maybe this very picture, since we don't see them. Or I would just say name 2 types of exocrine cells. So these ductal cells, as I mentioned, are secreting water and bicarbonate, and that's into the lumen. And then these acinar cells are secreting enzyme-rich. So the enzymes are what you're going to be studying on that last lab day, and studying in the digestive processes lab. So I helped you with that, and I will say, hopefully everybody's here now. We have 18 people here. I couldn't put it in Dropbox, which is probably why you couldn't find it, because my Dropbox account is full. I have to erase things. So instead, I put it on my Google Drive account, and I put a link into announcements. So let me know if you have trouble finding that, because you're going to need to know the names of many enzymes made here, some of which we're going to be using to test digestive processes or you're going to watch the videos where they're testing digestive processes. They're all going to be pancreatic enzymes that are being tested. So these enzymes are released by these acinar cells when CCK tells them to, and these guys secrete the bicarbonate when secretin tells them to. And since secretin stimulates CCK and CCK stimulates secretin release, they're always there together, which is nicely coordinated. These are exocrine because there is a duct, and they eventually go out the main duct to that sphincter of Oddi, or hepatopancreatic sphincter, and go into the duodenum, where they are going to act on the food. So here is our picture. If I go to our actual slides, which I may briefly, we can make this bigger. I actually took this picture to illustrate the endocrine cells of the islets versus the exocrine. So you can see the exocrine are kind of in these circular structures, which are those groups of acinar cells. These cracks are probably ducts, but even at higher power, I don't really see the cuboidal cells around them like you saw in the picture in the book. And then these are endocrine, so they're going into, they're secreting insulin and glucagon into the blood. Yeah, so I will end this part by just pointing out there are 3. These 2 are the, actually there are 4, because these 2 are together, but for some reason they're the same activity, but he filmed them in 2 different YouTube videos. This one's a really short one though. And so this is where you're getting data. You're getting it from this one, and this one. If you can do that, which would be ideal, before we actually meet on Tuesday, they're not very long YouTube videos, but you need to stop them so that you can write down and think about the answers for the handout, which is posted already in your folder in files. So here's what you should be focusing on. And we'll talk about these things next Tuesday. So there are controls that are set up, and we'll talk about why you have them and what the various types of experiments are experimenting for. Some of it's pretty obvious, like, they boil the enzyme and denature it, and then it shouldn't do its job anymore, which is what the experiment is testing for. So we'll come back to this on next Tuesday and go over it. And I think then it's due, I believe, Tuesday as well. So be prepared for that. Let me see, there's something in chat here. And I can't read chat until I get this. Don't think so, no. I don't know what you're saying, Augustine, but...
Someone else just asked something. Yeah.
Oh, okay.
Yeah, I don't have a question.
My private chat I didn't see, so, all right. Okay.
I had a quick question, Professor.
Yeah, no.
So are the liver and pancreas slides due today then? Because you didn't change it. I don't know if you were going to.
I didn't change it? Okay, I'll change it till Tuesday. So, and if for some reason they show up late, and as long as they show up Tuesday,
Okay.
I won't take any points off for lateness. Yeah, sorry about that. I probably meant to change that. That's a lot of slides too. All right, I'm going to, if it'll let me, escape this. Oh, I guess maybe I'll stop sharing and then it'll let me. Let's share the slides that you have. I left them open, I thought. Maybe they're open as another window. Don't save anything in there. Close that window. Here they are. Okay, so let's look at a few of these liver slides. That is the exact same one that you just saw. So here is another one showing you, I'm just pointing out there are a few other liver slides. So here's another one showing you several lobules in the middle of the liver rather than just that one on the edge. You only still have a central vein here. This is a big artery. That's not a central vein. It's in that edges. Here's another large vessel. So you can see the lobules are, surrounded by other lobules in this area. And I'll probably stop sharing. Okay, let's look at another one of these. Let me see if I do it this way. I think that's the same one we just talked about. So there are there's 3 or 4 of them that are 40X. You don't have to draw all of those, but I would look at them in case I use a different one. And the hepatocyte one is the one we already saw. It was the one I embedded in your slides. So pancreas. I think I only have one slide on the pancreas, the same one you just saw. If you'd like, you could make this, a lot bigger so that you can see the acinar cells here with their nuclei that are kind of pie-shaped around the duct. I just can't, even at this higher magnification, really tell any cuboidal cells and ducts like you see in that other picture. So remember, in the endocrine, there are alpha cells and beta cells, but like in 103A, I don't expect you to tell me which ones are alpha and which ones are beta. Just know each of, the alpha release glucagon and the beta release insulin. And I'll tell you a secret, there's actually gamma in there too, but I won't ask you that since the book doesn't cover it. Okay, looks like a couple more chats. Is histamine released by S or H cells? I thought there was S. Does the book say H? I thought they're called S cells.
I believe in the first lecture you called them H cells, but you later corrected yourself and called them S cells in a different lecture.
Yeah. Seems like they ought to be called H cells, but they're called S cells. Oh, you can't see this last photograph of the pancreas? Is that true, Alexis? Or, okay. All right, so I probably have to stop share and share again. Sometimes I can't tell whether it's letting me move around. So there's the endocrine part with the alpha, beta, and we won't talk about gamma cells. And then these guys are the cynar cells. But I honestly can't really see ductal cells like you saw in that published picture. So I'd have to use the published picture if I wanted you to identify the ductal cells. So that's what you're doing in the pancreas. Okay, if there are no other questions, I'll let you go, and I will change the due date right now. So that shouldn't be any issue. For Tuesday on the last of your slide drawings. And while you guys are working, I will be giving feedback on the slide drawings that are already turned in. So I can tell I have a pile of grading, but I'm going to try and do those first so you get some sort of feedback to relook at those slides if you need to. Okay, any questions? What exactly do we have to draw for the liver and the pancreas again? It's in the study guide, which I think I closed. Oh, Jesus, that's nice. I just opened the wrong thing. Good. Okay. And it's a PowerPoint, which is what I was going for. Okay. Oh, that's it again. Jesus. Okay. PowerPoint. Open the PowerPoint. I'm hoping that lab test 3. No. I guess it doesn't count me having opened that. I'll have to go. Maybe because it's in this. I don't know. I don't know why it didn't count that one. But here it is, the study guide. Let's go. I'll go to the liver and pancreas histology thing. I think there's not one slide that says this is what you label. Instead, you're right, it is a little bit confusing here. So instead, let me share this so you can see what I'm talking about. I just embedded it all next to the slides, things you need to know, like if I'm pointing at a structure like lobule, I would understand hepatocytes. So maybe I should make a list and put that in announcements because I didn't make a list like before. Draw 40X. I could do that now. But that's what I guess when I was thinking, I was helping you more by putting it next to what you need to know next to each of these slide pictures. So that's for the low power, and then the higher power, recognizing these triads and knowing what flows through them. And then for the ductal, you'd have to use this picture. And for our slides, the cynar and the alpha and beta cells, again, they're listed here. Is that something you need, do you think? Might I say 40X, draw at least this at 40X, label these things. Do you need a list like that? So you can answer me in chat if you do.
Yeah, I think that would be helpful.
Okay, so I'll put that in announcements. There's only probably minimally 3 drawings you have to make. You could edit 3 or 4, depending on whether you want your ductal cells and your cynar cells on the same slide as the alpha and beta cells. All right, so I'll do that right now, and then you can refer to that in announcements. Are there other questions? Close chat up so I don't see it right now.
I had a quick question about one of the slides we had to draw up the turn in today. It was about the rectal anal transition.
Okay.
There wasn't much to label on that one, right?
No.
Okay,
Yeah.
I just want to make sure.
Yeah, it was the tissue subtype change, which is the rectum, which is the anus. And I suppose knowing there are glands, but I'm not sure what you're seeing there are glands. I think they're just, because it's stratified squamous, so it's probably just folds. All right, any other questions? Okay, so I'll stop sharing and zoom over and make the list that I'm looking for the liver and the pancreas, okay? And so you know what to label. I probably have it from the last study guide in here already. So if you can turn those in by 5 tonight, the ones on the rectum and all that, then turn in the liver and the pancreas by 5 on Tuesday. And I'll put that in announcements too. Okay. So I'm here, and just give me a shout. Does anyone want to be in a study, in a breakout room before I leave this window and go off to the other window? Okay. All right, I'll first make the breakout room. Sign manually. Okay, sign. Okay, I think I've got everybody who wanted to be in that room. All right, and I will go make